Hello, everybody. Welcome to Mice Tech Talk with the Technical Information Scientists at the Jackson Laboratory. My name is Dolores, and we are here to serve the research community by answering mouse-related questions. This is one of the ways that we have to connect with researchers around the world. I'm very excited because today we are joined by my colleague, Grace Berryhill, who earned her PhD from UC Davis, studying mammary gland development and tumorigenesis in mouse and large animal models using histological, molecular, and bioinformatics approaches. She has extensive experience in cancer research and is very familiar with the PDX models and preclinical services offered at JAX.
Indeed. Thank you, Dolores. It's a real pleasure to be here. In today's episode, we'll be discussing a bit about what PDX models are, where you can find them, and how they're typically used in preclinical studies. So hopefully you're ready with your questions because it's time for our Mice Tech Talk today. So today, let's go ahead and talk cancer modeling with PDX mice.
Awesome. For those of you watching live, you can use the comment section of the video to tell us where you are viewing from. And if you have any related questions, please post them there and we'll try our best to answer them today. So Grace, let's start with the most basic questions. What are PDX mouse models?
Sure. Yeah. So PDX is the abbreviation for Patient Derived Xenograft. So essentially, this is tumor tissue that would be obtained directly from the clinic. And then that primary human tumor tissue is engrafted into immunodeficient mice. So extremely immunodeficient mice, non-skid gamma mice or NSG mice that are a permissive host for the engraftment of primary human tissues. So tumors that take from this initial engraftment can then be serially passaged and banked as an established model for oncology studies. And so part of the real power of cancer modeling with PDX is that it allows us to encapsulate more of the heterogeneity that exists in human tumors, and with the aim of really more faithfully modeling that complexity of human tumor biology in a preclinical setting. So they really are a pretty different system to work with than commonly used tumor cell line xenografts, which are also incredibly valuable and have a really important place in cancer biology studies. But PDX offers us a little bit of a different niche there.
That's right, and that's so important. PDX models are considered more translatable than using cancer lines, and this is because most cancer lines are propagated in long-term two-dimensional plastic flasks generally, and it has been shown that they are molecularly different from the original tumors because of the selection that they go under in non-physiological conditions. In the past decade, PDX models have been highly recognized and valuable as a valuable tool to understand cancer biology, to help in drug discovery, and to translate findings for optimal treatment of human disease. Really, no other system up to this point has proven to be more representative of human cancer. And that's because PDX models generally retain, as you were mentioning, the histopathological and clinically relevant molecular features seen in human tumors, as opposed to cell lines, which cannot be differentiated histologically. So do you have any more comments on this point, Grace?
Yeah, sure. I think one of the things that's really key is that PDX models do more fully model the heterogeneity that we see in human tumors. So this encapsulates differences between cancers with different primary sites. It also encapsulates differences in molecular signature that can exist within a tumor from the same primary site, right? Or And those molecular signatures can really strongly impact therapeutic strategies that are available clinically. And so what we have here is a system where we can characterize PDX models at the genomic and transcriptomic level in addition to assessing histomorphology in response to standard of care therapeutics. Really, I think one of the great values here is this enables researchers to make more targeted and tailored, model selection decisions when they're conceptualizing and planning their preclinical efficacy studies, based around specific parameters that they believe will be important for the mechanism of action of their therapeutic. So what we're able to do is be targeted in the models that we're selecting, and also within a system that is more representative as it is, primary human tissue that is passaged or engrafted in a mouse. So, it does get fairly complicated, I think, a lot of times in terms of kind of visualizing all the models that are available and selecting between them. But I think there's an awful lot of power in it. And one of the things that we'll spend some time on together today, I know, but that was also referenced in an earlier episode here, our Mice Tech Talk episode, my colleague Xing Tong discussed in episode 21, Let's Talk Databases. He gave us a very brief overview of where to find our PDX models in the collection at JAX. But Dolores, to go ahead and keep that conversation going, would you be able to walk us through our PDX database and the basics of how to refine your searches? I'll go ahead and see if I can share my screen to drive that there. Give me one second.
Right, let's try to share our screens. If you're looking at this episode from your cell phone, warning that the search will look pretty tiny in your screen, but we'll do our best to go and walk you through it. So that is what you're seeing. I can see your screen, Grace.
Super.
We are looking at the Patient Derived Xenograft. That's the search form, and you have the link in the YouTube description if you're watching by there, or in the links that we offer at the bottom of this episode in our website. So you can find that link pretty easy. So for this example, what we chose is to search for a PDX tumor, not based on the site, the primary site of the tumor, but rather based on a genetic mutation. And so let's look for a variant annotated in the BRAF gene. So we have to enter BRAF right there, and then show us all those PDX models that we have with dosing studies that have included BRAF inhibitor, specifically dabrafenib. So we had to click like we did there for dosing studies, and now we are searching for treatment with a BRAF inhibitor, dabrafenib. And then after we have that, we hit search, and we have a few models that came up. And those are actually 3 melanoma models that meet those search parameters. And Grace, can you please walk us through the information that we display for this model? For example, if you pick the first one, what kind of information can we find in this database?
Sure, I'd love to. It's actually a pretty interesting example that I encourage you, if this piques your interest, once we finish the episode, you can maybe re-engineer this search and have a look at the other 2 models as well, because it is a pretty interesting example to walk through. But what we get to when we click on the individual model detail page, or I guess I should orient you first, I'm operating based upon the model ID and clicking that model ID to take me to the model detail page for that particular model. So we can see a brief summary of the model. And then what I'd like to do is show you a little bit more about what we can see in terms of information about genetic variants. So I'll go ahead and come to the variant section and expand that out here. And then what we have is we have crosstalk between our PDX model database and the JAX clinical knowledge base, which is a pretty powerful tool to allow us to see information here about these annotated variants that exist in the tumor system. And so here we can see different genes, different variants in those genes, the variant effect. We can see the effect on the protein if that's known. And then we can see annotations that are predicting sensitivity or resistance, as well as potential treatment approaches. And so let's go ahead and zero in on. Did you have something to add in, Dolores? Sorry if I cut you off there.
No, I was just going to clarify something if people are not that familiar. So basically, if a gene has a specific variant or a specific variation of the sequence, you would expect that they would respond or not respond to certain treatments, for example.
Yeah, potentially, right. If that's known, if there's information known about, the actionability of that particular variant. So let's go ahead and zero in on our BRAF variant, since that was what we used as the driver of this search. And so here we can see that the variant that this model carries is the BRAF V600E. Variant. So what we can see here is that the predicted protein effect is actually a gain of function of BRAF. And so then what we can see if we carry this over, that a potential treatment approach would be a BRAF inhibitor. And so what I do know is that a BRAF inhibitor that exists is dabrafenib. And so that was also what we searched on for our dosing studies. So if we scroll down, we can have a look at the information that exists with our annotated dosing studies. You can see details about the design of those studies, numbers per groups, dosing, etc. And then we can visualize the results of these dosing studies in a number of modalities here. So let's go ahead and start by looking at, simple tumor volume over time with our different treatments. And so I'm going to zero in on our control and the dabrafenib. So again, that BRAF inhibitor specifically. So I'm just going to toggle off. So I'm just showing those two groups for clarity. And then what we see is tumor volume here on our y-axis, and then days of the study down here on the x. And what we can pretty clearly see with this particular example is that we do have suppression of tumor volume in response to dabrafenib, that BRAF inhibitor, which nicely aligns with the molecular signature that we could see in this particular model as annotated by that BRAF V600E mutation or variant, which does carry with it a predicted sensitivity from our CKB annotation. So it really does nicely illustrate how we can tie all of these pieces of information together. And so we can see that visualized in a spider plot. So we're looking at tumor volume over time for individual mice within these treatment groups. And so we can see, some variation, which I would say is absolutely to be expected when you're working with PDX engrafted mice. We see variation within our control as well as some within the dabrafenib treatment group here. But this looks like a pretty nice set of data that we can see here. We can see additional plots change into tumor volume as a percentage, as well as tumor growth inhibition, where we can see that with all of the treatments used in this particular dosing study. So again, I just wanted to give you a fairly brief orientation of how to navigate some of the information here. And I think just to illustrate how you can maybe conceptualize thinking about searching for models based upon markers that are important for you. And we're always here to help in the Technical Information Services group to be able to get you to a selection of models that may meet your needs. So I think if you'd like, Dolores, I can go ahead and stop sharing my screen unless there's something else I'd like to see.
Yeah, that would be great. Thank you so much for walking us through that. It's amazing the amount of information that there is, especially for a subset of our PDX models. I would like to take some time to check for questions that were posted by our viewers. We have a question from Janine, and she's asking, how do I find tumors from patients that are resistant to certain therapies? Do you have a take on that?
Yeah, it's a good question, and it's certainly one that we get fairly regularly. And I'd say resistance is something that can be a little challenging to address in its kind of most rigorous definition. What we can do is look in a couple of different places. We have some information from our models about the patient treatment history. So the treatment history of the patient that the PDX, that the tumor was taken from, that the PDX was derived from, which can tell us, a kind of a snapshot of information. It generally doesn't tell us response to therapy. It generally tells us treatment or not. Doesn't really give us a sense of response. What we can search on in the mouse models of human cancer database, where we search for PDXs for those models that do have dosing studies annotated for them, which is not all of the models. So right. We're a little bit limited there, but it's growing a lot. We can search based upon the define resist criteria, which can give us some information about the progression of disease in response to certain therapeutics. But it does get challenging to get to a place of specific questions of resistance. I'd say that can be a challenging one to address.
Right. I agree. We have another question from Monica. It says, do you see changes in growth rate kinetics in PDX models, I suppose, and sensitivity between passages? So as Chris showed in her dosing studies, in the example of dosing studies, I mean, there is definitely changes in growth kinetics. If you have a cohort that originated from the same PDX model passage, we definitely see differences in growth kinetics, and that's why a lot of times when people are preparing cohorts for these mice, a lot of times they do overage, they engraft a little bit more, just to make sure that within that time frame they will have enough mice with desired tumor growth to start enrolling on the study. So they don't have to wait until every tumor reached that point. I'm not sure if you have something else to add, Grace. You're more used to doing efficacy studies yourself.
Sure. So thank you for that question, Monica. It's a really good one to bring up about understanding differences between or really how to accommodate for passage number. And so just real briefly, passage number would refer to, the number of kind of generations, I guess, for lack of a better descriptor, that particular tumor was carried through the mouse. So the initial time you put a tumor in a mouse, the initial engraftment of the primary tissue from the clinic would be the passage 0 number. And then the next passage, so expanding that out, would be passage 1. You take from that, put it back into a mouse, passage 2, so on and so forth. And so that's really important to keep track of for the reason that, there may be differences that can exist in that tumor between passages. So one of the things that I mean here is, let's take, for example, breast tumor models that are screened for hormone receptor markers, estrogen receptor positivity, for example. There certainly are cases where that particular marker status can be lost over time. So one thing that I would say in regard to passage is to make sure that you're screening to verify that your particular marker of interest is retained across passage. And another thing that we strive to do is to provide researchers that we're working with relatively low passage number tumors, low passage number engrafted mice to use on studies, et cetera. So that's something else. But in regard to growth rate kinetics, between passages, I'd say, sure. I don't have a side-by-side example, so I don't want to overspeak on that. But we do know, for example, that if you're bringing a model that has been banked and exists in the cryobank, if you're bringing that out and grafting it into live mice to be able to establish again, the initial passage out of the freezer is going to grow pretty slowly. And then once you're engrafting fresh tumor tissue into live mice from there, you would have faster growth from there. So that I can say as an example of some differences. I don't want to overspeak. I suspect also some of this is going to be a bit model dependent as well. But I'd say the moral of the story is that passage number is really important to be aware of, and to for good practice, not mix and match in between an experiment, and to verify that your marker of interest is retained across passage numbers as well.
Great. Thank you for that, Grace. We are running out of time, but I would like to address Vidya's question about, are these tumor models primarily subcutaneous, and do we look at distant metastases as well? So yes, our models are subcutaneous in the flanks of NSG mice or NSG SGM3, depending on which tumor we are looking at. So we always do it that way, or at least most of the times we do it sub-Q. And routinely, we don't look at our PDX models and wait until and analyze routinely if they metastasized or not. This is something that, of course, you can do in your own lab, but we don't do that routinely.
Indeed.
So,
great questions, all. Thank you.
Very good questions. Very good questions. So we'll definitely have to make another episode of PDX, and I would like to talk more about PDX models and immuno-oncology, but we'll have to leave that for next year. Well, this is all the time we have. Grace, can you comment a little bit on the linked resources in this video so people know what to explore more?
Sure, absolutely. So certainly we provided a link to our PDX search, which we spent a little bit of time on together today. You also see information that'll take you to our preclinical testing services that our team offers here at JAX. We have extensive expertise working in these systems, as well as others for oncology and immuno-oncology preclinical efficacy studies. And then we also have a link to a webinar on predictive cancer modeling using PDX mice. So please do spend some time there. We're always happy to help however we can.
Perfect. So our next Mice Tech Talk is episode 26. Let's talk designing genotyping assays on Tuesday, December 15th. So you can go to LinkedIn and YouTube to watch it, and we look forward to seeing you there. So this is Dolores saying stay healthy, stay safe, and stay excited about your research. Bye, everybody.